Welcome to the deep dive. If you are a medical professional, maybe an educational content creator, or, just someone who believes that true mastery really starts with the fundamental how and why, then today's deep dive is for you.
That's right. We're setting aside the clinical algorithms for a moment and going straight to the source code of the human body, physiology.
We're extracting the highest yield, most integrated concepts from foundational medical texts. The focus today is on the principles that govern everything: cellular life, electrical signaling, hemodynamics, and volume control.
And our goal is to build a high-speed, comprehensive, but still really rigorous summary. Something you can use immediately, whether you're teaching others or just trying to solidify your own understanding.
Exactly. The mission is not just to list facts, it's to connect them. We need to prioritize the concepts that cause these physiological domino effects and link them straight to clinical reality. We're going from a single membrane protein all the way up.
All the way up to system-level homeostasis. Precisely. I mean, to understand a failing heart or kidney, you have to first understand the cell membrane. So let's start right there.
Okay, the membrane.
It's defined by its proteins. You've got your integral proteins, think G proteins or ion channels, that literally span the entire width of the bilayer. And then you have the peripheral proteins.
Which are more loosely attached.
All right, very loosely attached, often by non-covalent electrostatic bonds. They usually just sit on the inner or outer surface.
And how those cells connect to each other dictates how a whole tissue behaves, especially with permeability. Let's talk about intercellular connections.
Yeah, they function like these sophisticated gates. Tight junctions, or zonular occludens, they control what can pass between the cells.
And a critical point here, especially if you're teaching this, is that tight is a relative term.
Absolutely. They can be incredibly restrictive, a real barrier, like what you find in the renal distal tubule.
Or they can be, leaky.
Exactly, leaky, allowing significant movement of water and solutes, which you need in places that do high-volume processing. So the renal proximal tubule or the gallbladder.
And what about gap junctions? They're different.
Totally different. They're direct channels connecting the cytoplasm of two cells. They permit electrical coupling, which allows for synchronized activity. This is absolutely vital for, say, the coordinated contraction of the heart muscle.
Okay, so that's the structure. Now, movement. Let's look at transport across that membrane, starting with the simplest form, simple diffusion.
Right. No carrier, no energy input. It's purely passive.
Just moving downhill.
Downhill along its concentration gradient. The classic solutes here are small and nonpolar. Think oxygen, CO2. They just slip right through. Permeability drops off a cliff as a molecule gets bigger or if it has a charge.
But the second you introduce a protein carrier to help out, the rules change.
Right. Whether it's facilitated diffusion or active transport, you now have to deal with three governing principles: stereospecificity, saturation, that transport maximum, or TM in competition.
And for energy strategy, we split active transport into two camps. Primary active transport is the big one.
It's the big spender. It hydrolyzes ATP directly to shove solutes uphill against their electrochemical gradient. And the foundational example, the one everyone has to know, is the sodium-potassium ATPase.
The NAK pump. It's the workhorse of the cell, isn't it?
It's the physiological workhorse, burning up a massive percentage of the cell's energy budget just to maintain those gradients. And it has a strict stoichiometry.
3 sodium out, 2 potassium in.
Always. 3 Na plus out for every 2 K plus in. That maintains the low intracellular sodium we need for signaling and, of course, the cell's negative resting potential. And clinically, it's the direct target of drugs like digitalis.
We also can't forget the proton pump, the H plus AK plus ATP.
Oh, yeah, in the stomach. It's another primary active transporter directly consuming ATP to secrete hydrogen ions into the gastric lumen is what makes the stomach so acidic.
Then you have the more clever approach, secondary active transport.
It's very clever. It doesn't use ATP directly. Instead, it uses the potential energy that's already stored in an existing gradient, usually that strong sodium gradient that the NAK pump works so hard to create.
Which leads to two subtypes. You have symport or cotransport.
Right, where both solutes move in the same direction. The classic example is the sodium-glucose cotransporter in the kidney and the small intestine. Glucose gets a free ride uphill into the cell, powered by sodium moving downhill.
And if they move in opposite directions?
That's ANPort, or countertransport. The Na plus MK2 plus exchanger is the perfect example. It uses the inward sodium gradient to pump calcium out of the cell, which is crucial for muscle relaxation.
Okay, so let's pivot from specific solutes to water itself. Osmosis. When we're calculating osmolarity, we're just counting particles. But for effective osmosis, That's a different story.
It is, and this brings us to the reflection coefficient, sigma. This is, I think, an essential differentiator for any medical learner. A reflection coefficient near 1 means the solute is totally impermeable.
So it exerts the maximum osmotic pressure. It pulls water like crazy.
Pulls it powerfully. Think of big plasma proteins like albumin. They can't cross, so they pull water in.
Now, what about the opposite? A sigma near 0.
That means the solute is completely permeable. It just waltzes across the membrane, so it exerts practically no effective osmotic pressure. The prime example here is urea.
It's an ineffective osmoly.
Right. If you pour salt outside a cell, water rushes out. If you pour urea outside a cell, the urea just follows the water inside. There's no effective shift in water.
That osmotic balance is so key for cell volume, but to get to action, we need to talk electricity. Let's move into excitable tissues.
So the potential across a membrane is all about ion concentrations or permeability. We use the Nernst equation to calculate the equilibrium potential for a single ion.
Which is the exact voltage needed to stop that ion from moving.
Exactly.
Yeah.
And for a neuron or muscle cell, you just have to know the key values. Sodium's equilibrium potential, ENI, is very positive, around plus 65 millivolts.
And potassium's EK is very negative.
Extremely negative, around negative 85 millivolts. So what does that mean? It means sodium is constantly trying to rush in, and potassium is constantly trying to leak out.
So the driving force for an ion is the difference between the actual membrane potential and its equilibrium potential. And current only flows if a channel is open.
You need a driving force and permeability. And those channels opening and closing are what define the action potential. The rapid upstroke, the depolarization, is driven by the explosive opening of sodium channels.
Forcing the membrane potential up towards sodium's equilibrium.
Precisely. That's true for nerves, skeletal muscle, and the fast response cells of the heart.
And then repolarization is the trip back down, driven by potassium channels opening and K plus rushing out, pushing the potential back toward its own negative equilibrium.
This brings us to a really crucial comparison point: excitation-contraction coupling. The mechanism is fundamentally different between muscle types.
Okay, let's start with skeletal muscle. The action potential triggers calcium release from the sarcoplasmic reticulum, the SR.
And that released calcium binds specifically to troponin C. Now, normally the troponin complex inhibits the actin-myosin interaction. But when calcium binds, it physically moves tropomyosin out of the way.
It removes the inhibition.
It removes the block, and that allows cross-bridge cycling to start.
Okay, now contrast that with smooth muscle.
Smooth muscle is different. The calcium, which comes from both outside the cell and the SR, binds to a protein called calmodulin.
Not troponin.
No troponin here. The calcium calmodulin complex then activates an enzyme, myosin light chain kinase, or MLCK. MLCK phosphorylates the myosin heads, and that is the necessary step to allow myosin to bind to actin and start that slow, sustained contraction.
That's a fascinating switch from electrical to mechanical. Okay, let's shift gears to the plumbing, the cardiovascular system. And the key concept here is Pois's law.
Specifically, the radius component of that law. Resistance is proportional to 1 over the radius to the 4th power.
R is proportional to 1 over R to the 4th.
Which is just a staggering relationship. It means if you have the radius of a vessel, you increase the resistance 16-fold. This is the physiological leverage point.
And the arterioles are where this happens.
Exactly. They are the primary site where the body makes these tiny adjustments to radius to control massive changes in vascular resistance and therefore blood pressure.
And while flow is normally smooth or laminar, sometimes it gets chaotic. We get turbulence.
Turbulence creates brutes, those audible vibrations you can hear with a stethoscope. Clinically, this happens when blood velocity gets really high, like downstream from a stenosis, or critically, when blood viscosity drops.
Like in severe anemia.
Yep, low hematocrit means less viscous blood, which is more prone to turbulence.
Okay, before we leave the heart, we need to revisit the electrics. We have 2 types of cardiac cells.
Fast response and slow response. In the SA node and AV node, the slow response cells, the upstroke, phase E0, is caused by a slow inward calcium current.
And they have automaticity.
Right. They spontaneously depolarize during phase 4 because of that funny current, the IF current.
Yeah.
Now contrast that with the ventricles and Purkinje fibers, the fast response cells.
There, the phase U upstroke is driven by a fast inward sodium current, just like a neuron. But their defining feature is that long phase 2 plateau, which is sustained by a slow inward calcium current, balanced by an outward potassium current.
This brings us right back to the clinical action of digitalis. It has a positive inotropic effect. It increases contraction force.
But the mechanism is indirect, and it's so beautifully integrative. Digitalis works by inhibiting that foundational NAC-A ATPase pump.
So if you block the pump, intracellular sodium starts to build up.
It rises, and that diminished sodium gradient then cripples the function of the sodium-calcium exchanger, that antiport system we talked about earlier.
So the exchanger can't efficiently pump calcium out of the cell anymore.
Exactly. So intracellular calcium rises. More calcium means more available to bind to troponin C in the cardiac muscle, which boosts the force of contraction. It's a stunning cascade from one tiny transporter to global heart function.
Okay, before we hit the kidneys, let's nail down the high-yield autonomic receptors. You just have to know these cold.
You do. Alpha-1 receptors use the GQ protein pathway, leading to IP3 and calcium release. They primarily cause vasoconstriction. Prazosin is the classic antagonist.
Beta-1 and beta-2 both use the G's protein, increasing SEC-MP, but with different effects.
Right. Beta-1 is heart-focused, increases heart rate contractility. Dobutamine is an agonist. Beta-2 causes smooth muscle relaxation. Think bronchodilation, vasodilation in skeletal muscle. Albuterol is the textbook agonist for that.
And don't forget the nicotinic receptors, specifically the NN type in the autonomic ganglia.
Right, they're ion channels. And they are blocked by hexamethonium. But an important distinction, hexamethonium does not block the nicotinic receptors at the neuromuscular junction.
Okay, shifting to the renal system. It's all about fluid management. We measure body fluid compartments with markers.
Yep. Total body water, TBW, uses things like tritiated water. Extracellular fluid, ECF, uses stuff that can't get into cells, like mannitol or inulin. Plasma volume uses dyes that stick to albumin, like Evans blue.
And the intracellular fluid volume, ICF, is always calculated indirectly. It's just TBW minus ECF.
Right.
And the really functional part of this is understanding water shifts. Let's run through the three classic scenarios.
Okay, first, isosmotic volume contraction. Think severe diarrhea. You're losing fluid that has the same concentration as your plasma.
So ECF volume drops, but osmolarity doesn't change.
Exactly. No osmotic gradient, so ICF volume stays the same.
Okay, scenario 2. Hyperosmotic volume contraction. This is like severe sweating or diabetes insipidus. You're losing free water.
So your ECF volume drops and your ECF osmolarity skyrockets. This high external osmolarity acts like a powerful osmotic drag.
Pulling water out of the cells.
Right, so your ICF volume drops too. The cells shrink.
And finally, hyposmotic volume expansion. The classic cause is SIADH.
Syndrome of inappropriate ADH. You're retaining excess free water, so ECF volume goes up, but the osmolarity plummets.
And that dilute ECF drives water into the cells,
leading to an increase in ICF volume. The cells swell. Understanding those three is just essential for volume disorders.
Okay, now let's bridge a knowledge gap that will be critical for our final exercise. We need to talk about aldosterone.
The key mineralocorticoid. It acts mainly on the principal cells in the collecting duct of the kidney.
And what does it do there?
Aldosterone cranks up sodium reabsorption in exchange for potassium and hydrogen ion secretion. So high aldosterone means you save sodium and water, raising blood pressure, but you lose potassium and acid.
And the flip side is critical. Aldosterone deficiency means you lose sodium, causing hypotension.
And you retain potassium and hydrogen ions, leading to hyperkalemia and acidosis. It's a vital integrated pathway to know.
Okay, one last renal concept. Clearance.
Renal clearance. It's the virtual volume of plasma that's completely cleared of a substance per unit time. The equation is C equals U times V over P.
And substances like inulin or creatinine, which are only filtered, their clearance is our best estimate of the glomerular filtration rate, the GFR.
Right. Finally, let's touch on the bedrock of acid-base compensation. In metabolic acidosis, the primary problem is low bicarbonate.
So the lungs compensate.
They compensate by hyperventilating. That deep, rapid, small breathing you see is the body trying desperately to blow off CO2 to fix the pH.
And for metabolic alkalosis, the primary problem is high bicarbonate.
So the compensation is the opposite. Respiratory depression. You hypoventilate to hold on to CO2 and bring the pH back down. And if the patient's measured compensation doesn't fit the rule, you know you're dealing with a dangerous mixed disorder.
Wow. We have just raced through 4 major systems.
Find it.
It's amazing how the rules of a single membrane transport system dictate everything that follows.
It is. The sheer density of these integrated mechanisms is staggering. From electrical potentials to muscle contraction, vascular resistance, kidney function, it's all connected.
And the highest level of synthesis is understanding the controls. You have the CNS and autonomic nervous system regulating global factors like heart rate. But when you zoom in on specific organs, local control often takes over.
Absolutely. Think about the two circulations that cannot tolerate any interruption in flow, the brain and the heart.
Right.
Cerebral blood flow is incredibly sensitive to CO2. If your PCO2 rises, your cerebral vessels vasodilate powerfully to wash it out.
But the coronary circulation is different. It's not about CO2.
No, it's primarily sensitive to oxygen demand and hypoxia.
Mm.
The body's most potent local signal there is adenosine. When the heart muscle is working hard and getting hypoxic, it releases adenosine, which forces the coronary vessels to open up immediately.
Knowing those distinct local controls, that's the difference between a textbook answer and real clinical competence.
Exactly. So to tie a lot of this together, here is a final reinforcement exercise for you. It requires integrating the renal, endocrine, and cardiovascular physiology we just reviewed.
Okay, let's hear it.
A patient presents with 3 key findings: severe hyperkalemia, metabolic acidosis, and hypotension from volume contraction.
Okay, hyperkalemia, metabolic acidosis, hypotension.
We just detailed the role of a specific mineralocorticoid in how the kidney handles sodium, potassium, and hydrogen ions. Based on this physiological picture, which renal hormone deficiency would explain this precise combination of symptoms?
That's a great question. It forces you to use the underlying mechanisms, the specific transporters in the renal tubule, to solve a clinical puzzle. Go back through the facts and see if you can connect the dots.